Hello, and welcome to Hawk Mountain Sanctuary's Stay at Home speaker series. Today's program is Mapping Our Knowledge of Raptors Population Genetics with Marianne Gousy-Leblance. Hi, Marianne. Thank you so much for joining us today. My name is Jamie Dawson, and I'm the Director of Education at Hawk Mountain Sanctuary. Thank you to our wonderful audience for joining us today for this webinar. As you may know, Hawk Mountain is the world's very first refuge for birds of prey, and we continue to work hard to be leaders in raptor conservation, science, and education, locally and globally around the world. Hawk Mountain is a private nonprofit, and membership is the lifeblood of our organization. To all of our members, thank you so very much for your continued support. And if you're joining us today and you're not a member, we hope that you consider becoming one in the future. Hawk Mountain hopes that everyone remains safe and healthy during these times of COVID challenges, and we are thrilled to offer our local and global community a variety of free virtual programming. As always, Hawk Mountain greatly appreciates and depends on donations. Just so everyone is aware, today's program is being recorded. The video will then be accessible on Hawk Mountain's YouTube channel as a continued resource. We also have a link on our website directly connecting you to our YouTube channel. At any point during today's program, viewers may submit questions through the Q&A feature on the Zoom platform. We've designated time at the end of the program to take some questions from the audience. And we are delighted that Marianne is joining us today all the way from Canada to share with us her fascinating research on raptor population genetics. Before we get started, I'd like to take a moment to share some of Marianne's experience with our audience. Marianne is currently a master's student at Moncton University in New Brunswick, Canada, in the Canada Research Chair in Polar and Boreal Ecology Lab of Nicolas Lecomte. Her master's project is in collaboration with Dr. J.F. Terrien of Hawk Mountain Sanctuary and Glenn Yannick from Savoie-Mont-Blanc University in France. For the last 3 years, Marianne has been working in the Arctic each summer, monitoring bird populations. She developed a strong interest in the Arctic ecosystems and on its avian species. Thanks to her previous undergrad experiences, Marianne had skills in population genetics that she decided to exploit for a project on an Arctic breeder, the snowy owl. She's also interested in bird migratory strategies and movement in addition to population genetic structure. Today, Marianne will present the first chapter of her thesis-based population genetics in raptors in general. Marianne, what inspired you to study raptors?
I think that raptors are just so fascinating and we have so much to learn from them. When I was in the Arctic, I could spend hours watching Falcon or snowy owl from far away.
Wonderful, wonderful. So how did you become involved with Hawk Mountain Sanctuary?
Yeah, like you said, GF is one of my advisors on the project, and he has access to many samples that I'm using on my snowy owl chapters.
Awesome. Well, we are so glad you are part of the Hawk Mountain extended family, and we are all looking forward to hear more about your amazing research in the Arctic. So Marianne, I'm going to turn it over to you.
Perfect. Okay, so let's talk about our favorite bird, the bird of prey, on a population genetic point of view. Like we all know, raptors are located all around the world. So here is a distribution map of the number of raptor species per country. Darker color, the purple, represent countries with higher number of raptor species. Lighter color, so the light green and yellow, represent countries with a lower number of raptor species. One thing that we can see from this figure is that the majority of the country with a high number of raptor species are more located in the southern hemisphere. Raptors are important in every ecosystem as they play many roles. For example, they are an indicator of biodiversity. As top predator, raptors are located at the top of every ecosystem. An ecosystem with a high number of raptor species or individual will be considered with a higher biodiversity. Raptors can also serve as indicator of environmental health. An ecosystem in good shape should have more raptor individual or species than an ecosystem in bad shape. Raptor can also serve as cultural symbol in many countries, and they can also play regulatory role. For example, they can limit pest population. For example, an owl feed on small rodent that can eat our crops or any other culture that are important to human. So owl can limit these species. But with the actual anthropogenic activity like habitat destruction, wind farm, pollution, or hunting, there is dramatic consequences on raptors. Indeed, there is about 18% of all raptor species that are now considered threatened with extinction, and a little bit than half of all the species are exhibiting a global population decline. The decline is more important in vulture group. Vulture represent about 80% of all the threatened species. On the graph here, we can see how vulture are becoming threat in the last few years. In my example, the vulture is the African vulture. Because all of that, we really need to increase and refine the current toolbox for the conservation of raptors all around the world. It is why today I'm going to talk about one of these tools, that is population genetics. You can ask me, what is population genetics? It is the factor and variable that explain the difference between individual and population. So how can we study population genetics? We can study by studying the genetic variation within species and among population of the same species, but also evolutionary factor that explain this variation. In my example here of the small cartoon, it is an example of an evolutionary factor. The beak of the bird is changing, evolving, depending on the resources that the bird eats. You can also ask me, why should we use population genetics as a tool for conservation and management? Because population genetics represents species sensitivity and adaptation capacity. So how a species can react to a change in this environment. For example, if its main prey is changing. Sensitivity and adaptation are two key elements of assessing vulnerability of population. So how population can adapt in time, how they're gonna survive. In other words, population genetics can help us predict abundance and future distribution of any species or population. It can also inform us on the genetic and demographic response to a species, to a change, like if they suffer from habitat destruction. All of these elements are important for conservation and management of endangered species, like Madagascar fish eagle, that is one endangered raptors. One important concept in population genetics is the structure. So what is a structure? It is a population that has a spatial structure. So the distribution of its genetic pattern is not uniform. In my example here, the genetic pattern is the small owl. So we have the green owl and orange owl. In my population here, they are distributed uniform in space. So we can consider that there's no genetic structure. But if I have something like that, where we are really seeing a cluster of orange individual and a cluster of green individual, we can say that there's a structure. And if we zoom in, we can see, for example, that there's a mountain chain that limit the movement of our individual. So in my example, 2 cluster of individual, so there is a structure. The last concept that I'm gonna talk about today is the effective population size. That is key to any conservation genetic practice, as it informs us on the demographic trend of population and species. So we can know if the species is increasing, decreasing, stable, or suffer from a bottleneck. Effective population size can inform us on the actual demographic trend of the population, but also the past demographic trend. It's also give us an idea of the sustainability of the population if the current effective size is maintained in the future. We can also use that to assess any effectiveness of genetic management. So my project that I'm gonna talk about today is that we want to assess the current state of knowledge and gap within on population genetic in bird of prey, but also suggesting where future research should go. And we're gonna do that with a systematic review. Systematic review is used to know what has been done in the past, but we can also highlight the trend in the gap in a fieldwork. It's not available for raptors, so this project is the first one assessing the population genetic in raptors. So we did a literature survey through 2 online databases, and we limit our search to 2019 to make sure that we have similar method in the article found. We look for every scientific article assessing population genetics in raptors. Here are an example of the scientific article that we found. For each of these publications, we record a bunch of information to try to answer of our question of trend and gap in population genetic in raptors. So we record the species, the family, the localization of the study, the International Union of Conservation of Nature status. So it is a standardized conservation status that is used for most of the species. We also record the population trend, the sample size, the state of sample. We can get DNA from a live bird, but also from dead bird or museum specimen. We record if they compute an effective population size, the DNA sources, and if they found a population structure or not. For our result, we found about 72 scientific articles assessing population genetics in birds. That include 48 species. So that represents only 9% of the species that have been studied. On the graph here, we can see a phylogenetic tree of the raptor investigated. A phylogenetic tree represents how species are close to each other. In our study, the species investigated include 5 families. So we have the hawk and eagle, the falcon and kestrel, the owl, the vulture, and the osprey. We didn't find any article assessing caracara bird or secretary bird. If we zoom per family, here is the graph that we can get. So for the hawk and eagle, there is only 10% of the species that have been studied. So that represents only 24 species on the 236 existing species. For the vulture, there is only 14% of the species that have been investigated. So only one species, that is the Indian condor. For the falcon, there is only 11% that have been investigated. For the osprey is by himself in this group. So we have one study assessing the genetic of the osprey. And finally, the owls are the group with the lowest percentage of investigated species, with only 6%. We can conclude from this graph that there's a few species that have been studied in every group. It is less than 20% for all the groups. For the localization of the study, here is the map that we can get. So one thing that we can see here is that the majority of the studies are located in North America or in Europe. For the rest of the world, we have no information. When we link that with the real abundance of per country, here is the map that we can get. So here is the percentage of genetically investigated raptor per country. Darker color, the purple, represent countries with a higher percentage of raptors investigated, and lighter color represent countries with a lower percentage of raptor investigated. So here we can clearly see what I was telling you, that except North America and Europe, for the rest of the world, we have no information. And when we come back at the map that I showed you at the beginning, when I told you that the area where there is a higher number of raptor species is the area where we have no information on the genetic of these raptors. For the conservation status, here are the scale that the IUCN is using. So at one size, we have the extinct status. So it's species that doesn't exist anymore, like the Reunion kestrel. And on the other side is the least concern status. So it's species that are doing great. So in our study, there is about 58% of the species investigated that have the least concern status, like the common kestrel. For the near threatened status, we have about 17% of our species investigated, like the spotted owl. And finally, for the threatened status, include the critically endangered and vulnerable bird. There is about 25% of our investigated species, like the crowned solitary eagle. If we link together the conservation status and the family, here are the graphs that we can get. So we have the number of study per each group. So the hawk and eagle are the group with the higher number of publicity on them. It is also the group with the higher number of species with a conservation status. The two species with the higher conservation status in this group are the Madagascar fish eagle and the Ridgway hawk. For the falcon group, there is only one species investigated that is endangered, and it is the Saker falcon. And finally, for the owl, there is the snowy owl that is endangered, and the blackish-tongued fish owl that is endangered. Sorry, the snowy owl is vulnerable, and the blackish-tongued fish owl is endangered. For the population trend, there is about half of the investigated species that exhibit a decreasing population trend. For about 31% of the investigated species that showed a stable trend. The decreasing is more important in the owl group, with 60% of the investigated species that exhibit a decreasing population trend, and 54 for the hawk and eagle. So if we link together the conservation status, the localization, and the population trend, here is the map that we can get. So the upper map represents the number of threatened and declining raptor per country. Once again, darker color, the pink, the purple, represent countries with a higher number of declining raptors. Lighter color represent countries with a lower number of threatened and declining raptor. One thing that we can see from this figure is that the majority of the country with a higher number of threatened and declining raptors are located in Asia and in Indonesia. If we go to the lower figure, here it is the percentage of threatened and declining raptor investigated. So darker color, country with a higher percentage of threatened and declining raptor, lighter color, lower percentage. We can clearly see again that the only place where we have information on the threatened and declining raptor, it is in North America and in Europe. For the rest of the world, no information. And in Indonesia and Asia, where there's a higher number of raptors, no information. Finally, quick result. For the sample size, there is about 231 individuals per study. The majority of the sample came from a live bird. Only 22% came from a dead bird or museum specimen. Only 18% of the study compute an effective population size. That is very low according on what this variable can give us. And finally, most of the study use non-destructive, so non-invasive sampling, so buccal swab, blood, or feather. Non-invasive sampling is more used because it's fast and easy, and we can get plenty DNA to do our genetic analysis. For the feather, we can use malted feather, so feather that we can find, for example, next to a nest, or we can use plugged feather during bending, so we can use breast feather or back feather. For the feather, we are always going to use the lower part of the feather to extract DNA from. On the picture here is an example of buccal swab sampling on a juvenile falcon. This technique is also easy. We just need some saliva out cell to extract DNA from. Finally, for our result on population genetic structure, there is about 78% of the study that detect genetic structure with an average of 2.32 population per study. So what can we do with a population genetic study? Here is an example on the region eagle owl in Spain. Here, they wanted to know if the habitat fragmentation that they are seeing is also creating population isolation in this species. So one of the results that they found is that there is 3 population units in Spain. So we have the darker red that is located more on the east part of Spain. We have the light pink that is more central Spain. And we have the beige one that is located in this southern part of Spain. From this figure, we can think that there is no population isolation because there's still movement between the 3 populations, as there's still individuals that have similar pattern from other populations. If we look at the population on the east part, the dark red, there is still some beige and some pink from the other population. So no population are isolated, so the species should be okay. Our principal conclusion is that a very small percentage of raptors have been investigated so far. Most of them are geographically biased toward North America and Europe, like we can see on the map here. Also, most of the species are with a lower level of trap. But good news, we are seeing an increase of non-invasive sampling. The next step should be first to increase the number of published studies on population genetic in raptors. Also, with the increase of resources, analyze technology method, there is a real potential to do new study connecting traditional monitoring data with genetic data for raptor. For example, we could link tracking data, so GPS data, with genetic data to investigate migratory behavior or migratory divide. But we also need to increase our tool for communication between scientists and people that are doing the conservation work to make sure that the knowledge is really applied. For my part, then my step should be my second chapter that is focused on snowy owl. So we're gonna look for the population genetic of snowy owl in North America. Like I told you earlier, snowy owl are vulnerable. They are also in decline in North America. So we really need to increase our knowledge to understand how we can conserve this species. Finally, if there's one thing I want you to remember today, is that population genetic is an essential tool in raptor conservation. And we need to increase the number of studies, but also the number of species investigated. So I would like to thank my advisor on the project. So Jeff from Hawk Mountain, Glenn and Nicolette. And thank you, Jamie, to having me today.
Thank you, Marianne. That was such a wonderful presentation. Thank you so much. Wow, and now if anyone in the audience has any questions, feel free to type them in and we can go through your questions. And what an impressive, and we have a comment coming in saying excellent talk, so great job, Marianne. What an impressive amount of scientific literature review. What was the most challenging aspect of this research project so far?
I think it was first to find the article, because when we are looking for a keyword, like we start with about 300 article, and like we have to read the abstract and the title and try to narrow it down to papers that are really in our topic.
Yes, wow, wow. So, and when are you scheduled to complete your master's work?
Should be in September, but we will see.
It's coming up. All right, we have a question. Oh, nope, it's a comment saying, wonderful talk, Marianne. Yeah. Thank you. All right, here is a question. How did you compare the quality of the different articles? Were there some that you questioned how well it was done?
Usually when the article I publish, they are reviewed by other scientists than the people that did the paper. Usually we can trust the quality of the paper when it's published.
True. Thank you. Thank you so much. Well, I don't see other questions coming in at the moment. We'll give it a few seconds to see if there's any more. Have you had any restrictions with your research at all based on the COVID?
I was supposed to go back on the field in the Arctic this summer, but COVID, like all the north part was closed, so no field work this year.
Aw. Okay, we have a snowy owl question. How did you
Okay.
genetically sample snowy owls?
I have access to many feather that came from bending. So usually people, when they bend owl through like snowstorm project, that was one of the project that is involved. I can get feather from that. So we have the sex, the age, and the localization of the owl. So I have a bunch of samples that come from feather. And for the rest, it's mostly veterinarians. When they find a dead snowy owl, they can sample it for us too.
Thank you. Okay, we have another question. How many samples are needed for genetic analysis?
Usually it depends on the markers that you're going to use, but about the mean in the review, it's about 200, but if you have a good marker resolution with about 100 sample, you can get nice result.
100. Okay, wonderful. Thank you. Marianne, thank you so much. It was a fantastic presentation. I know that I certainly learned a lot, and we hope that in the future we can see you again at Hawk Mountain Sanctuary. That would be wonderful. And thank you for sharing everything with us. Thank you to our wonderful audience for joining us. Oh, wait, let me see if this is a question coming in. Oh, here's a question. This is a good one. Marianne, what is your favorite bird of prey?
I love the snowy owl, but I also love the burrowing owl. They are so tiny, so they could be nice to study too.
Oh, yeah, actually, one of our former trainees studied burrowing owls down in Florida earlier this year, and we did a webinar on it. Oh, in the spring, I think. So, someone is sharing. They wanted to share from Henry, who's age 10, wants to share that his favorite is the Falcon. So thank you, Henry, for sharing that.
Oh yeah, they are amazing. Like last summer, I had the chance to see one, and it was like white. It was amazing. Yeah, a big male. It was so cool.
Wow, wow, wonderful. So again, thank you. If there's any more questions popping in, just shoot them in and I'll read them. So again, Marianne, thank you so much. Thank you to our audience. We are very excited at Hawk Mountain because guess what tomorrow is? Marianne, do you know what tomorrow is at Hawk Mountain? It is the first, it's the first day of our migration count. So we're really excited. We're having a migration kickoff and we're doing a lot of things to celebrate. We have nonstop activities from 11 a.m. to 3 p.m. And these activities are all free with a trail passes purchase, and of course, always free for members. So we're going to have throughout the day live Raptors up close programs where you'll get to meet a hawk or perhaps an owl, all safe and social distance in our lovely, stunning, brand new amphitheater. And we also have other programs going on throughout the day, such as Name That Raptor to work on your ID, some ID tips. We also have Hawk Mountain history going on at South Lookout throughout the day, as well as a naturalist in the garden, in our native habitat garden. So it's going to be a wonderful day. We hope to see you on the mountain, if not tomorrow, sometime. So, and as always, we have more virtual programs coming your way soon. So this is what's in store. Next Wednesday, the 19th, we have Totally Terrific Turtles and Tortoises at 1 p.m. Next Friday, August 21st, we have Raptor Conservation in Nepal with Dr. Tulsi Subedi at 10 a.m. Wednesday, August 26th, we have Custodians of Our Ecosystem, Women, Raptors, and Unsettling Words with Dr. Diane Husick, and that's at 7 p.m. Thursday, August 27th, At 11 o'clock a.m., we have Sanctuary Storytime, Fidget's Folly. And finally, on Thursday, August 27th, at 4 o'clock p.m., we have Living the Island Life, Cara Caras, with our own David Barber. So lots coming up. We hope to see everyone soon. Marianne, again, much gratitude to you for your fantastic presentation. And I hope that everyone has a great weekend. It's Friday. Woo! Bye for now.